Hi, my name is Savannah Fouch, and today, along with Catherine, Julian, and Ria, we will be discussing the nulliviruses in the families Ifilridae and Partitaviridae associated with the common eastern firefly, Photinus piralis. Now that was a lot of words. Let's summarize it really quick in this introduction. So essentially, the question that these researchers are asking is, can fireflies carry viral infections? And today we'll be discussing the common eastern firefly, which is present in North Carolina. When the sun sets in the fall, you'll see some fireflies in your backyard if you look hard enough. The biological question that will be discussed today is, what is the viral diversity associated with populations of the common eastern firefly? And could these viruses play a role in the decline of firefly populations? It's essentially asking, can these fireflies carry viruses, and does that negatively affect their populations? And the hypothesis of these researchers in this publication is the common eastern firefly hosts viruses from the Ephoridae and Partitaviridae families, and these viruses are present at intermediate frequencies in natural populations of fireflies. That essentially is saying that these researchers, before doing their research, is thinking these fireflies can carry these two viral infections, and they are present in different populations of fireflies throughout the state.
Hello everyone, my name is Julian Giuseppe and I will be reviewing the methods used in this firefly experiment. So first off, what was done? The scientists in this study wanted to find out if fireflies carry viruses we didn't know about before. To do this, they used different tools and methods, kind of like detectives gathering clues to solve a mystery. Here's what they did, step by step. So, these are the 5 methods they used. The first method was finding the viruses, or in their terms, metatranscriptome sequencing. Or in simpler terms, the scientists reviewed fireflies' genetic material, RNA. RNA is like a tiny instruction sheet inside your body that helps make proteins which help you grow and stay healthy. And they use this to detect unknown viruses, like recording sounds in a room to identify songs. And this correlates to image number 1, which illustrates a microphone recording different voices in a crowded room, with speech bubbles labeled firefly RNA, virus RNA, etc., to show how scientists listen for viruses in genetic material. The second method, checking infection rates, PCR screening, they tested wild fireflies using a sensitive method, PCR, which is a method used to make many copies of a specific piece of DNA from a sample, to see how many of the fireflies were infected. And this correlates to image number 2, which shows a magnifying glass over a row of fireflies, with some marked as infected and others not infected, representing how PCR detects viruses in samples. The third method, rebuilding virus genomes, or genome assembly, is when, using computer programs, they piece together virus genetic material, like a puzzle, to get the full virus blueprints, or the virus makeup. And this is illustrated in image number 3, a torn-up book page being put back together like a puzzle, symbolizing how scientists piece together virus genetic material. Method number 4, sorting data, and they used special software which helped compare the viruses by matching them to known ones. And this is illustrated in image number 4, showing a computer screen displaying a matching game, where unknown viruses are compared to known ones, showing how scientists identify the viruses. And finally, the final method, understanding virus relationships, or phylogenetic analysis. And this is when they created a, quote, family tree of viruses to see how the new ones were related to other insect viruses. And this is shown in image number 5, which shows a family tree, but with viruses instead of people, illustrating their relationships over time. Thank you.
Hello everyone, my name is Catherine Cummings, and I'll be talking about the results and interpretations of this study. After using metatranscriptome sequencing, which gave researchers a map of what was in a specific type of code called RNA, they concluded that there were certain viruses there that came from other virus families and not in the particular family that infects the firefly. Next, they did DNA sequencing, in which they created another map of another type of code called DNA, which is a precursor code to RNA to look for similarities between the code in the fireflies and existing codes of viruses in storage. From this, they found similarities with two families of viruses called Inflaviridae, which normally infects mosquitoes and ticks, and Partitiviridae, which normally infects plants and fungi. Once they found this out, they called the virus from the Inflaviridae family Photinus pilaris inflavirus 1, and the virus from the Partitiviridae family Photinus pilaris associated partiti-like virus. They wanted to find out how much of this virus was present in fireflies. They used another test called PCR to replicate the exact segments of DNA that were associated with the two viruses in each firefly. They found that 7 out of the 13 fireflies sampled had the Photinus pilaris inflavirus 1, and 5 of the 13 fireflies had the Photinus pilaris associated partiti-like virus. As I mentioned before, 2 unknown viruses were identified and named as new species. After the initial realization that there were viruses of the Inflaviridae family and the Partitiviridae families in the codes of the firefly, researchers decided to do what's called a phylogenetic analysis of proteins made from DNA to look for patterns and similarities between the discovered viruses and other viruses within their families. It was determined that the Photinus pilaris inflavirus 1 proteins were only 28.8% similar to their relatives. As mentioned earlier, the virus itself was found in 54% or 7 out of the 13 total sample of fireflies. For the Photinus pilaris associated partiti-like virus, it was 54.3% similar to its relative virus proteins and was found in 38% or 5 out of 13 of the fireflies sampled. This information gave researchers enough information to conclude that these new viruses were a new species. So to know about these new viruses could actually help researchers and conservation experts work together to keep the firefly population going. Thank you, everyone.
Hello, my name is Rhea Wu, and I will be presenting the conclusions and future ideas. So what is the main conclusion of our micropublication? Well, the study identified 2 new viruses in the common eastern firefly. What the researchers did first to come to this conclusion was they found the virus's RNA to assemble a complete and big collection, and they saw how common the virus was in the firefly population. And for comparison, we can think of a government collecting fingerprints of its population, and then when it compares fingerprints that are very similar to each other, they can notice the frequency of these fingerprints. Secondly, through phylogenetic tree, researchers determined these viruses are their own species. And how did they do this? They saw that the most recent common ancestor of these 2 viruses with the other viruses was very, very old. So let's say that I'm comparing my brother and I. We would have a very most recent common ancestor because that would be our parents. However, if I were to find the most recent common ancestor of a classmate of mine, it would be much older than my brother and I's most recent common ancestor. The researchers' consistent detection of these viruses meant that they're regularly found in fireflies. This study shows how valuable a compilation of RNA or genetic material from a big population can be for understanding viruses in insects. What are the future ideas of the study? What else can we do with the results? We should also research the effects of the virus on firefly health and behavior. For example, we know that flus make humans sick. So we want to observe whether or not these viruses will have an effect on fireflies like the flu does on us. Secondly, we should track across populations and environments to see how these infections spread. For example, we could put tracking collars on all the dogs in Chapel Hill, and we would get multiple populations and multiple environments because these are different species of dogs that are spread out over the entire town. Third, we should also research to see if there's a link between viruses and smaller firefly populations. In other words, we would be observing if viruses kill these fireflies in large numbers, just like how the Black Death killed much of Europe's population.